Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
It's my great pleasure to welcome Adrian Lee from University of Washington to talk about brain dynamics. This is one of the topics that probably will capture a little interest in a lot of research here doing neural network type of research. So Professor Lee graduated from MIT Harvard Speech and Hearing PhD program. He recently moved to UW just a few months ago.
A year ago.
A year ago. Oh, wow, time flies. Okay. I got the opportunity to visit his lab a couple of months ago. There was a lot of exciting things going on over there. So today he will come here to tell us something about speech and hearing research as related to brain imaging and as related to brain dynamics. So I'll give floor to Adrian. Okay.
Thank you. Thank you, Lee, for the invitation. It's great to be here. My goal today is to try and convey what type of, what's the frontier in neuroscience, and really in terms of the interface between neuroscience and engineering. I think that's actually where the most exciting part is. So where we're at and where we want to head to. My home department is speech and hearing, and I'm an adjunct professor in electroengineering. But our lab is actually based in the Institute for Learning and Brain Sciences, and we'll show you the type of equipments that we have for capturing brain dynamics over there. But I want to start with something that sort of maybe capture our thinking every day, the combination of neuroscience and engineering, and what are the things that the challenges that we face. Well, one of the things that we're interested in is how do people selectively attend to a sound of interest. This is something that a human can do quite easily, but a machine yet can do properly. Another thing is recognizing speech. Just a brand new speaker coming in, a human can potentially understand even my weird accent of mixed with colonial British, Australian, and some Bostonian accent. Hopefully you can still understand my speech properly. You don't need to train yourself for 20 hours of of tokens of me speaking. So how do we do that? It's something that we want to capture in terms of how our brains do it and learn from it too. One of the things that we're also interested in is whether we can design machines, and now it depends on different levels, whether we can mimic, interact, or even complement human abilities. So understanding where we're at in the brain, what are deficits, can we capture brain signals and interject? And of course, this is the ultimate goal, right? Have a seamless human-computer interaction, and I think that's something that you're all interested in. So truly believe that a successful interface between neuroscience and engineering would, although it actually starts needing from the brain state classification problem. Can we identify which brain states you're in, and therefore make brain-computer interface? And maybe in having artificial intelligence that's actually enhance our capabilities. So let's start with our basic problem, right? One of the things that we study is the cocktail party effect. Now, in this environment, there are lots of different conversations coming on, right? And for us, it's really easy to pick a certain conversation. So for people that are at UW, they might say, hey, I've got a Huskies ticket. Can you come over and do you want to join me for the weekend to watch it? For grad students or postdocs, they probably say, hey, there's free drinks in room 204B, right? You've sharply tuned to that. And that's defined back in 1953, identified as the cocktail party problem, yet we can't solve it computationally right now. I'll tell you why this is also important from a learning environment point of view. Here's a Chinese lesson for the kids, right? It says, ni hao. And repeat after me, and Jenny says, ni hao. That's great. Unfortunately, if you've ever taught, that's not the environment, but this is, right? So being able to selectively attend affects education, affects a lot of things. This picture also reminds me of that, there's a lot of machine learning going on here, right? The interesting part is that these are the little machines that learn every single day. In terms of iLabs, they actually, we do a lot of brain imaging on how they learn. We want to understand how they do it so that we can better learn. So in some sense, this is a generic problem statement of how we can learn from the best learners. Okay, so if the signal is not good, even though it's ni hao, poor Billy might have got mao, which is very different from the signal. So if you don't attend it properly, ni hao means how are you, mao means cat, right? So if you don't selectively attend and fish out the acoustic signals properly, there will be information loss. So even though it's a very perceptual question, it has a very high upstream consequences. So it's really hard to describe what an auditory object is. So I'm just going to play a game with you here. We're going to play the game called Password. You tell me what the password is. Okay, listen to the male voice and tell me what the password is. There might be a prize at the end. So male voice, give me the password.
The
I'll buy you a
password
drink if
is
you stand
Huskies.
up now.
Okay, so what is the password? Huskies.
The password is Huskies.
Okay, now I said that there might be a prize at the end, right? So what did you
I'll buy
miss
you
out?
a drink.
Now listen to what you could have done to get that prize from me.
The
I'll buy you a
password
drink if
is
you stand up
Huskies.
now.
It is a very expensive game to play if I miss out, right? But I can direct your attention to the same physical stimulus, yet you can pick up the signal depending on what you listen to. This is the dynamics that we talk about. Attention is rewiring your brain at 100 of milliseconds level or giving you the respective cues to bias the physical stimulus so that you get one message out or the other. That's cocktail party effect. And that's what an auditory object is. We listen to the male voice versus the female voice. Now, it's slightly different from the visual scene analysis, right? So in vision, here's a screen in front of the whiteboard. And in that case, something is in the foreground, occluding the background, hence there's an edge. And edge detection in vision helps a lot to segregate objects in front and background. In auditory, it's very different. We call it transparent scenes. Spectral temporal elements add on top of each other. And it's like transparent. It's really hard to segregate them. Now, there are different cues that we can use. Here, I'm using a visual analogy here and have different things in different colors. So you could imagine that I can attend to a certain feature. Here, if I'm attending to yellow, in terms of neurophysiology, we know that if you're attending to a certain color, those type of neurons actually are active more and therefore have a bias in terms of attention. Now, you can selectively switch from one cue to another. And also, I just played a game with you because I was fiddling around with audio, told you a lot about Huskies and UW. I primed you that the word is Huskies. So priming also works. So in our lab, we're interested in auditory cues, how they group sounds, and how to group acoustic sounds, and how selective attention works. So what are the auditory cues that we normally use? In an orchestra, you can listen out for the violins versus the flute, right? So that's timbre, intensity. I have a louder voice than the fan that's actually humming in the back. Now that I've directed you to that fan, you're probably just listening to that fan instead of me. I'm going to concentrate more on spatial cues and pitch. Spatial cues are nice, especially when you have beamformers in terms of, I know that you have a speaker, conference phones, you can actually beamform to a certain speaker. It's the same things that we use for spatial cues in human. Pitch, whether I have a high pitch, low pitch, male voice versus female voice. So just briefly, I know that auditory spatial cues, just going to talk about it briefly. So here's a subject's head. Here's an acoustic event coming on. And the sound arrives to the ipsilateral ear, the ear that's closer, slightly, ever so slightly faster than the one that's opposite, by tens or hundreds of microseconds. That's good enough for us to have something called the interaural time difference cue. We human can actually go as far low as 10 microseconds as that discrimination. Interaural level difference. The head at higher frequency acts as a caster acoustic shadow, such that the closer ear actually receives the signal slightly higher by a few dB than the signal that's further away. And that gives you the acoustic cues. There are some spectral cues as well. Given all this, now I can direct you to listen to one cue versus another. What we want to do is to find the brain dynamics that's associated with you rewiring your brain really briefly to listen out to a stimulus. And here's the paradigm that we use. We ask the subject we cue the subject which side to listen to. So here's in left arrow. And then they maintain fixation. And then what happens is a stimulus comes on, two simultaneous digits. One's coming from the left, one coming from the right. One has a high pitch and one has a low pitch. And so because the arrow was in the left, pointing to the left, the right answer when we ask you to respond would be 3. Right? Now, you could imagine that I can just rewire your brain slightly differently using the same stimulus, but point the arrow to up and down. So now pay attention to the pitch. The pitch of this, I've denoted high pitch in red. So it's if I had the arrow going up, you listen to that simultaneous speech again. And to answer correctly, you have to listen to the high pitch, and the correct answer should be 2. And what we're going to do is once you've been primed what to listen to, look at the brain dynamics to how you actually rewire to listen to that cue. And so that's the dynamical process that your brain goes through. The technology that we use is as follows. MEG measurement. This is newly installed in UW 2 years ago. It's a multi-million-dollar machine. I think the price tag is in the inventory is $3.4 million, I think. Sits in a room that is magnetically shielded. It's at 306 channels measuring the magnetic fields coming out of your brain. We simultaneously use EEG, electroencephalography. So not only picking up the magnetic fields, we're picking up the electric potential on your head. On a separate session, we do an MRI scan. We want to know the anatomy inside your brain so that we can relate the brain signals that you measure and spatially co-register it such that we know where anatomically it's coming from. Once you do that, when you co-register your anatomy with your brain activities that you measure, what you can now do is to capture the brain dynamics. And we make brain movies. So I told you that they get a visual cue in the beginning, right? So here's the brain movie showing the first 300 milliseconds. I have to explain this brain a tiny bit. It's a funny brain view. Everything gets sort of inflated. I've marked the visual area, the auditory area, attentional area, and executive region.
So the color here indicates what?
The red spot is the flow of activities, brain activities. Precisely, this is MEG. So this is measuring about the postsynaptic potential. And if you look at the time scale, we can get down to millisecond precision. Right? So here, at 1,000 milliseconds prior to the onset of the sound, you receive a visual cue. It has about 100 millisecond delay to reach into the visual cortex. And as you see, there's a wave of activities now in your occipital cortex, in your visual cortex. Marching further down, now you're holding fixation, but you're actually now rewiring your brain to attend to either the spatial cues or the pitch cues, right? And you see there's a sustained activities in the attentional areas. This is actually called the frontal eye field. It's also involved in vision, directing your eye gaze from one position to another. If you think about moving your eye gaze, is casting a spatial attention from one position to another. And the question that we ask is this space, is this region, even though it's only for eyes previously defined, is it also useful for auditory spatial hearing?
Is that commonly recognized region called attention?
Yeah. So this is actually the precentral sulcus. Yeah. Okay. So at 0 milliseconds, that's when the sound comes on. So just watch the auditory cortex when it receives the auditory sound. Now, this is fundamentally slightly different from fMRI, right? Traditionally, in neuroscience, fMRI or even in terms of before that lesion studies, people talk about where in the brain matters, right? Here, we're trading off a tiny bit of the spatial resolution, but we want the brain dynamics, right? You know that in fMRI, every 2 seconds you take a brain picture, and you infer where things are in the brain. 2 seconds is an eternity in the brain, I hope, right? I can do a lot in 2 seconds. 1001, 1002, you just switch off because you know that 1002 is redundant. I'm counting 1 second, 2 seconds. Your brain shuts on and off depending on the context and all this stuff. So we want to capture at millisecond level what the brain is doing. Now, using this technology, what we can then do is very precisely work out the difference between your wiring of the brain even before the onset of the sound, as you're rewiring to listen to it. So right before the onset of the sound, when you're paying attention to space, the spatial cues, one part of the brain, frontal eye field, is involved, more active. When you're paying attention to pitch, another region, it turns out that this region, the superior temporal sulcus, has previously been recognized as the people using musical pitch discrimination and absolute pitch. People with absolute pitch shows stronger activities here. So naturally, your brain is recruiting different brain regions at different times to help you to do the task. Because of the fine resolution, and this is where we differ from fMRI, we can distinguish what happens right after the sound onset. Also, fMRI is really noisy, right? If you have ever been to an MRI scans, it's constant like that. It interferes with the very signals that we're measuring in speech and hearing. MEG is quiet. So we can definitively work out during the stimulus how we're reacting to the sound. And here we can actually see that while the spatial areas, the frontal eye fields, is still involved while the stimulus is on, it's less active or it's no longer differentially recruited when the sound is already on. Now, there are other ways of using this technology. Here we're sending an attentional probe into the sound. So what do we mean by that? Well, we're also going to give you two sounds, one coming from the left, one coming from the right. And your task is to count how many E's. So let's say, listen to the stream coming from the left. E, E, M, A, E, L, K. Great, there are 3 E's, respond 3. However, we're going to tag this with a certain frequency. A low, it's high frequency for the brain dynamics people. It's low in terms of the speech people, right? 35 hertz, 45 hertz. It's nothing that you think of as something that's high frequency. In the brain, we think of 4 to 6 hertz, 4 to 8 hertz as low frequency, or even down to 1 to 3 hertz. So in any case, we're tagging 35 to 45 hertz. We can talk more why we chose those frequencies. So basically, here's a speech signal, and we're going to amplitude modulate it. We're going to use that as a probe to find out where in the brain is actually responding to the 35 hertz or 45 hertz. Remember that when I cue you to listen to the left, this is now in the foreground, right? And if it's true that you're modulating what you're listening to, the things in the background shouldn't matter, right? You're concentrating to the left stream, and we can look at the tag signal. So what happens? Now, listen to the left, 35 hertz is the tag. Let's tune the brain at 35 hertz. We're just doing a phase lock value here, right? You can see that, again, the left frontal eye field, it's locked to the stimulus. And also the auditory cortex. Remember, 45 hertz is the side that you don't listen to, right? We told you not to listen to that. When we tune into 45 hertz, that signal is no longer there in the frontal eye field. You can counterbalance in which side that you're listening to, what frequency. Only the left frontal eye field is locked to the stimulus that you're listening to. And the probe, turns out psychophysically, is transparent, as in the subject doesn't even know that even if we change the probe in midstream, subjects don't even know. So it's a neat neuroscience cue that we can use, and also that can be utilized for a lot of eventual deployment for brain-computer interface.
Yeah, so that spatial resonance seems to be pretty good here.
Yeah, that's because we combine the anatomical scans with MEG and EEG.
Oh, okay. So after you do this computation, how does it compare with the MRI in terms of resolution?
Let's say fMRI, the canonical resolution is roughly 1 millimeter, although after a lot of spatial smoothing, it's around 5 to 6 millimeter effectively. We would say 1 centimeter. So for us, we care about the network approach. We don't care about whether this region, sub-region of the auditory cortex responds more to sound, pitch, or space. Right? We want the global picture of the brain. Eric Larson actually here, if you want to learn more about this experiment, I'm just going to briefly describe it. One of the things that we want in a lab is to eventually work out how to tune a hearing aid dynamically. Right? So hearing aids right now don't work in a very noisy environment. If you ever had older relatives listening, in a restaurant, they would rather take it off, right? That's because everything's being amplified. It doesn't selectively amplify to a sound, right? Now, if we want to selectively amplify the sound, we need to know where you're attending to. And one of the questions that we also need to address is, have you just switched attention from one person to another? So here, we're designing an experiment just to see whether you've switched your attention from one speaker to another. Use the same cue, whether you listen to the left or right, visual cue. 2/3 of the time, say, great, just respond to the 2 digits, one coming from the left, one coming from right, from the side that we've cued you on. But 1/3 of the time, we're going to say, oh, no, just kidding. I'm going to switch attention to the other side. Right at that 600 milliseconds when we told you to switch your attention, where in the brain helps you switch that attention? Turns out that the right temporoparietal junction, here's the manipulation point, right? Just kidding, switch attention. Green is switch attention, white is hold attention. You see a massive difference in terms of that particular region. There are other regions that's involved, the right frontal eye fields or the middle frontal gyrus. Great, there are different regions, but for us is whether we can capture that signal and do classification to work out whether you've just switched attention or not.
Yeah, but that area is normally not related to hearing.
Yeah,
so are you saying that the attention is got a generic attribute?
That's right.
That goes across audition and vision?
That's right. So one of the questions that we have is attention supermodal, right? If you're paying attention, it doesn't matter whether you're paying attention sound or space, it should be just an attentional network. Auditory attentional network has not been studied thoroughly, and that's what we want to contrast that to vision. Our TPJ has been implicated in visual cortex, and I'll show you. But the coolest thing, though, is this. Not only that we find a difference in terms of the brain dynamics, we then correlate back to the behavioral performance, right? And you see a massive correlation, depending on whether you can switch attention versus not, and correlate that into the differential activities of that region. It's almost like when Eric showed me this graph, I said, that's too good to be true. It's really highly significant. Clinically, we can use this to perhaps diagnose central auditory processing disorder. There are people that have normal audiograms, but just can't do things in a cocktail party environment. So from clinical perspective, this is useful. For machine brain-computer interface usage, it turns out that one part of the region is really good predictor of your brain state. And this is what Lee was pointing to, is that here's a visual attentional network that's been previously mapped out. I've shown you left frontal eye fields, right frontal eye fields, right temporoparietal junction. I think I'm a bit biased. I'm an auditory person. Auditory always cares about time and frequency. It is not the native axes that the visual people think about. So the question that we want to now address is, yes, we have a network. How do these signals pass from one node to another? Also, do they have this, when they pass the signal, can you tell that they have some correlation? How do they pass the signals in the first place? And I'll get back to that. So, as Lee said, we've sort of started my lab over here at UW and looking specifically into auditory brain sciences, but we have a neuroengineering goal in the lab as well. So I'm going to walk through different experiments, different people doing different things, and hopefully, if you're interested in it, please also talk to them during the course of the day. So Ross Maddox is also here. He is currently studying audiovisual binding problems. Now, why do we care about audiovisual binding? There are many objects that comes on, right? If you have a computer passing a video scene, right? How do you know whether the sound and the vision is the same person, right? It turns out that we use a lot of temporal coherence, right? At the syllabic rate, my mouth moves at this 4 to 7 hertz signal, and my syllables also coming out at 4 to 7 hertz. That gives you a cue that my mouth moving and my sound coming out, it's coming from the same object, right? And so we're designing experiments where Ross is looking into the different ways of binding, right? How temporal coherence can change your perceptions of binding. And the implication is that if we can harness this, it can also be turned into how machine can use that type of information for binding information. I think one of the main problem is auditory scene analysis, visual scene analysis is still a very hard computational problem right now. Can we learn from human to do that task? Another project that we have is with a graduate student with Elliot Saba, and he's co-supervised by Professor Les Atlas. And what we look at is the signal processing in the brain. Now, what does that mean? Speech people would tend to think about carriers at pitch, 100 and few hundred hertz, and then you have formants at the 1,000 hertz. As I alluded before, in the brain, we talk about 4 to 8 hertz as some brain signals carrier, and there's some high frequency at about 30 to 150 hertz. Here is showing a mouse moving in a maze, and where it goes into that particular part of the maze affects where the phase procession, when that particular, when they're at that particular region, that neuron is fired slightly earlier in phase of that carrier frequency. So it becomes that the phase of another frequency can be used to code here in where in space in the memory, in the hippocampus. People have done experiments on memory retrieval, items retrieval. If I give you a sequence of 1, 2, 3, 4 items, you can be locked into the different phase of a lower carrier to give you that sequential results back. So we're starting to analyze the signal processing in the brain, the coupling of different frequencies, and try to map that out. You can also imagine that this can be used eventually in EEG and MEG, right? I can dynamically look at how different parts of the brain are communicating using these lower frequencies, and therefore infer what brain state or what items that you're retrieving. I've mentioned this problem statement that our lab's goal, one of our lab goals is to get a hearing aid that can dynamically tune to the signals of interest. And in this case, we want to know what you're attending, but in a generic problem statement is what is your brain state. If we can measure that you just switch attention to one from another, I can also measure whether, oops, I've just made an error. Here's an error signal coming from the anterior cingulate cortex, for example. So a broader statement is, can we classify brain states? So Eric Larson, who's sitting in the back as a postdoc in our lab, he is working on this particular problem. We start with brains, lots of brains. Let me take a step back too. In terms of brain-computer interface, you can classify all your brain signals all you want for a particular subject. You will do very well. However, when you have a brand new subject, it's really hard to predict the n+1 subject what that brain state classification is going to be. We want to make a difference in that room. Can we predict a brand new subject what sort of brain classification taken into account of the bank of brain signals that we have? And this is how we are approaching this problem. First, we calculate the gain matrix. What this means is that we have an MRI scan we decimate the space in brain. This is pure Biot-Savart laws, just to work out how the sense is seeing each of the dipoles in your brain. And then, however, unfortunately, this is, we're talking to Lee before, and we're using the cheat data right now. We're doing modeling because we can simulate brain signals. Ultimately, we are never going to measure individual neurons in the brain, not yet anyway, right? Instead, what we
several neurons, let's say 30.
That's right, but not the whole brain. If you poke the whole brain, I'm not sure that organism will survive, right?
Technically, there are electrodes to get the signal from a single neuron.
Oh, that's true. Yes. I think we really want to do a non-invasive imaging approach. I think that's where the commercialization aspect will come in. I'm not sure everyone will put up their hands for sticking needles into the brain. So what we're measuring is something coming out of the head. It's the MEG and the EG signal. To infer back to where those signals are coming from the brain, you need inverse approach, right? This is high dimension, not as high dimension, rank deficient, need an inverse approach. So we do the inverse imaging, L2 norm constraint. We talk about the actual methods later on, if you're interested. One other problem. Turns out that, luckily, I guess, each of our brain, it's different. Look at each brain here, right? Each brain is just slightly, ever so slightly different in the geometry. They all have some resemblance to a generic brain, but they are all different. The difference in the folding changes the physical properties of your EEG and MEG signals by the right-hand rule, right? So a lot of brain-computer interface right now uses the motor strip, the motor central sulcus, and it happens that they all realign quite likely like this, right? It's nice. Once in the prefrontal cortex, where the cool things are happening, right? Attention, executive control. Those are the things that the brain signals that we really want to capture. The folding is so different that can massively change. Just imagine you're folding, right? Just do your right-hand rule here. The field will change very differently. So one thing that we have to do is to align all these little brains. Sorry, big brains, whatever. Spherical morphing is the approach that we use. Look at one brain, we're going to map it to a generic brain and come back to it just to see. So you can see that there's some sort of decimation, right? Low pass characteristics, but it captures mainly the so-called gyro pattern, which is important for us to get the field patterns. By doing so, what we can then do is to start approaching solving this N+1 problem. If we have a new brain, we're just going to cheat one more time. When we do the brain-computer interface for the brand new subject, all we need to do is just ask this person to get an MRI scan. So we're almost getting all the information. We don't need so much training of the classification. The goal is, by understanding, just capturing the anatomy of the brand new subject, can we do much better in terms of brain-computer interface? And so in a preliminary modeling experiment suggests that the answer is yes. Where is the regime that we do in general in terms of brain-computer interface when we want a massive deployment, right? It's low trial counts. We're not going to get the subject to do 100 trials and classify, right? That's the commercialization aspect is sort of low. We want low SNR, low trial counts. Those are the things that we want. And it turns out that in this simulation, having the anatomy of the untrained subject, right? So we're training all the brain data that we have with the anatomy. Now with a brand new subject, with that new anatomy can that actually help. And in the low trial counts and low SNR, it actually provides improvement. In fact, if you only just use the sensor space, so only classifying brain signals using the EEG channels, you can't even start projecting to the new subject. It normally doesn't work unless it's a well-defined motor imagery. There are few experiments that work, and that's actually where the mainstay of BCI right now. Okay, I'm going to conclude with a cool new experiment that's done by Nima, a good friend of ours. He's a former student of Shihab. I know that some of you know Shihab as well. Here is a direct recording on the brain. So this person's skull has been removed, electrodes placed on the brain. This is for epilepsy detection, pre-surgical planning. And the subject is being played two sounds. Again, it's the cocktail party problem. This is the CRM corpus, coordinate response measure, and the two sentences going on. And speaker 1 says, "Ready, tiger, go to green 5 now." Speaker 2 simultaneously, just like the example that I played you, says, "Ready, Ringo, go to red 2 now." The person is to respond to where Ringo went to, right? So when the subject listens to Ringo, great, okay, I need to know it's red 2 and ignore green 5. This is the computational problems that the brain is facing, right? You're all familiar with computational auditory analysis, try to fish out noise. It turns out, as an engineer, there's signal to noise ratio, right? There's always noise. In human, there's no noise. It's what I'm attending to, and what I'm not attending to is noise. It's really hard to define noise in human, because you can switch your attention at any point in time. So this is the signals that you get. It's the summation of the two. And if I'm only recording one signal at a time, this is the brain response, the spectral temporal response in the auditory cortex, to speaker 2 alone, to speaker 1 alone. This is the result of the unmixing of the auditory cortex due to the attentional bias to when you're listening to speaker 2 alone, speaker 1 alone, and see the correspondence here.
So how do they process the signal here? You get suppose you get n different electrodes.
Yeah.
The signal, do they do some signal processing?
Yeah, so the spatial dimension here, it's not the most important part. You can even tune down into...
electrode.
No, this should be multiple electrodes.
How do they mix them together? Is there any way?
So
that's very tricky thing to do.
Yeah, that actually gives you another piece of information. The weighting of the electrodes may give you a clue of where the actual area of the brain that's most responsive to this. So that actually gives you the where question, right? Here at least gives you the spectral temporal dimension, that the auditory cortex...
Weighted some, or do they more complicated process?
I believe so. I actually need to reread that paper.
Okay.
Yeah. But here's the sort of, a proof of something that we already know, right? We can do this. It turns out that the auditory cortex, when you have attentional bias, can modulate the spectral temporal signals like this. So I guess to conclude, I want to sort of open up of where we think that it's ahead and the road ahead of us, and also how that integrates to perhaps things that are maybe in 5 to 10 years commercializable, or a decade later. I think speech perception is fundamentally something that is time critical. Speech and hearing science, always time, frequency, axes are the most important dimensions to us. So one thing that we should start looking into is how different brain regions coordinate at different times. That should provide us with a new way of thinking in terms of the computational modeling. It's not just the where, it's not just the anatomical connection. People have been doing that a lot, right? How information is being passed dynamically. That adds the time dimension, even though we've, from the engineering side, we've done that using the hidden Markov or whatever hidden time state changes, right? You try to learn that. Well, why don't we learn from the brain as well, right? Another thing is the interaction between top-down and bottom-up attention. It is really hard to ignore when a phone comes on right now. Right? That's like an auditory siren. You automatically switches to the phone. And in fact, that's why a phone rings, is to grab your attention. Right? That's the competition between top-down attention. Oh, wait, I'm supposed to be listening to the speaker or not. And to ignore that phone ring. That's the competition. Now, a question that would be of interest is auditory siren an all or nothing? Right? Is a siren only just grab your attention like this? Or can it be sort of like a attention grabbing, but depending on your brain state, whether that will grab your attention or not? So I understand that there's a great interest in Microsoft looking into triage alert system, like the busybody system, right? In that sense, you want to know whether I want to alert the user, whether it's a good time to stay away from my work, right? Now, the decision point, of course, is based on a lot of data learning and how likely it is from the decision model to alert to the user. But you can flip it to the other side, right? I want to know how likely the brain wants to be alerted, or, hey, brain, I'm going to give you about 30% chance of actually capturing this signal. Just like if you have a green light on your phone, right? And you actually, if momentarily I'm just bored with my work, I see a green light flashing, I can make that decision myself too, whether I want to grab the phone and look at that message really quickly, right? So here, it's giving an option of not just an all or nothing alarm, but give the user back the chance to say, hey, here's a signal at the back. Oh, yeah, I'm actually interested in it because I just got bored a tiny bit, right? So it's an interaction between the computer and the user, and using that from a psychoacoustics end, how to integrate that in. Now, of course, there are a few other things that we can do, right? One, imagine in 10 or 20 years' time, when EEG technology is available such that we don't need all these gels, right? In fact, dry caps being produced now, it's slowly becoming more of a mature technology. You could have EEG caps that literally just have these micro sensors in your hair, picking up the signals, wirelessly transferred to your phone device. The key is, what signals are you picking up? And what are the key signals from the brain that is usable to interact with the device? That's the key, right? And that's why we're interested in brain state classification. We want a bank of vocabulary of your brain such that we know how to look it up. Moreover, to look it up is one thing. We need to know the dynamics. The other is how generic it is, right? We don't want to train every user all the time. Maybe someday, by using anatomy, we can just say, hey, here's out of the box a new phone device. Here's some EEG caps. Just put it on this way, and we'll do the learning myself. So that's where I'm going to end, and just like to acknowledge in terms of funding, Lee was asking what type of funding agencies now are interested in brain state classification. From a hearing aid point of view, the National Institute for Deaf and Communication Disorder, so sort of the hearing research branch in NIH is obviously interested in the hearing aid application, but the defense is also starting to get into the brain state classifications for reasons that might be obvious. And I'd like to thank my collaborators, both in UW, in Boston University, and also at Mass General. And if you want further information, here's our website. Thank you.
So going back to this attention, sort of inverse, with inverse engineering, probably just mentioned about. Do you know any computation model that somehow can give you similar kind of the output that they measure, so that the same kind of processing technique afterwards can recover one of the stream?
The two questions here, right? One is, what dimension should we use, right? So what feature space, what hyperspace should we do that classification for computational auditory analysis, segregation, auditory stream segregation? I think what we haven't been using, and this is the type of research that we want to at least give clues of what auditory cues we use, right? So grouping cues are the most important things. Continuity of sound. So it's there's a whole branch of auditory grouping literature.
The way you show the result, right? You don't even have to care about all the cues. As long as you can produce something that gives you the same kind of measurements, maybe similar measurements.
This one, right?
Yeah, this one I'm talking about.
No.
No?
The brain that you measured really cares about the cues.
Really? So if you
The person that is doing it is using all the cues that the person can use to segregate this, or else this sound wouldn't work.
So somehow you have to get the cue right in
Yes.
order to do this.
And the interesting point was we were also talking about a metric of how good you are in terms of after you segregate, right? It's good to have an L2 norm, right? A mean square error, engineering standard go-to. That may not be the most relevant thing for the user, right? A mean square error might have spikes in there, which really perceptually unpleasant, right? Just like MP3 coding, why is it that transparent coding works? You need to work out from the MP3 is from the perceptual side, from the cochlear side, you can work out the masking. Here is the higher level, right? What grouping cues can make the streaming more seamless, and you can still have some residual error that is irrelevant in terms of getting a stream out.
So if you go now a few slides here. Okay. Yes. So the input now we know it's a mix of the signal. Right?
Yes.
So there is actually a missing diagram that I really want to know in terms of the raw signal that you measure.
I see.
I mean,
from an
electron.
electricity, yeah.
And then you have those.
Yeah.
And then whatever process they do, they will recover this. Assuming that attention is all applied in the brain, right?
Absolutely. But don't forget the following, right? We're measuring from the auditory cortex, right?
And then you ask the patient to focus on one of those.
Right. But I've showed you in previous slides that when I pay attention to space or pitch, it uses different areas. So here it's there's a spectral temporal modulation of the physical stimuli, which is great, but that doesn't necessarily tell you where the attentional signal is coming from. Somewhere else in the brain is telling the auditory cortex modulate this way, right? So it is the trade-off between you want to know locally very well in terms of the spiking of the neurons versus I want a global view of the brain and how things are being connected. And I think you need both. There's every single level in neuroscience is important, right? Down to single unit neurophysiology, you have multi-unit recording, and for us, it's a system neuroscience approach.
But obviously, I think the significance of this piece of work is that it actually shows you that whatever recording you have, recorded is sufficient for you to get the original. So what it means is that if you, in terms of research, right, if you have this computational model, maybe they can be verified through some, kind of experiment, and that gives you reasonably good output that you might measure over here.
Yes.
Then it actually shows that you can actually recover the original. That actually allows you to investigate what cues are important, or otherwise. That you do modeling to figure out.
Right. And you could imagine that if I drop some spectrotemporal element, would that matter? So there are some metric that you can use, getting what the brain cares about versus not.
And so far, no idea about how attention is injected into.
We're working on that one.
Yeah, okay.
What would happen if you did the same test on attention with subjects that have a severe attention deficit disorder? I work with a student who has a severe, so I'm looking at teach you about the time.
Yeah, there are a whole host of psychiatric disorders that may influence attention, ADHD and also autism. We're talking about groupings here, right? How different frequencies in the cochlea, even though you hear pitch coming out, they all have to fire at the same time and group all these things, right? So back in the Terhart's day, talk about pitch templates, right? Frequency templates. But ultimately, it's a grouping binding problems. How do I bind these things together from different frequencies to all the different dimensions? It turns out that autism is characterized as a lack or a hypoconnectivity. The long-range connectivity is not as good. Now extrapolate, it's probably a problem with grouping, or at least some aspect in the ASD spectrum is the lack of grouping. That could also be in ADHD. We don't know. And there's a whole host of questions stemming from the perceptual grouping onto sort of a higher learning disorder. Yeah.
Are there significant differences between gender or age? Is their ability to discriminate, to focus attention? I chew my wife out all the time.
I really want to do that study without being fired, but we don't know. We don't know. We haven't systematically studied it. That's the official. Yeah.
So clues are fairly high on the processing or understanding of the clue. Have you tried working with the lower level clues that you influence the subject even without him knowing that?
Yes. So let me rephrase this. This is a high-dimensional feature space that you're looking at. There is space, there is pitch, there's all these things. You can tune into whether you can attentionally modulate one special cue, which might be stemming from brainstem if it's ITD or ILD or other cues. We're actually doing an experiment right now, just trying to see whether there is a direct coupling from your eyes with very rudimentary binaural cues coupling. I think traditionally, speech and hearing sciences stem from the engineering side. Ever since the 50s, 60s, it's always bottom up. As good engineers, anything that top down is just noise. Let's cut it out and understand the bottom up very well. I think in the last 5 to 10 years, we're starting to try to see what the top down signal is. So there's not much literature out there. So we have to systematically go back all the way down to brainstem, all the way down to cochlea to see where it's top down effect. And that's actually hard to measure.
Something about that. We've also done experiments where, in addition to playing these stimuli, we put in these short, quiet noise bursts that users don't even realize are there. And if you look at their performance on the task, don't affect their ability to selectively attend to one thing or the other. But then when we look at the brain activity, we see that there are areas of the brain that are specifically involved in suppressing the response to these sort of distracting stimuli. So we're trying to work out what the network is that allows you to do that.
So even if you don't know that it affected you.
Yeah, that's after you've been primed.
Well, the users don't even know that they're there, but yeah, they're doing some task in this distracting sound. And we asked them afterward if they even noticed it was there, and every subject said no. And yet there's this brain activity that's involved in allowing them to not notice it, essentially.
Yeah.
Let's see if I can raise the question. So it sounds like this attention process is kind of generic, but is it true that it really all happens kind of at the top, or is it, or like if you measure what's going on in the auditory pathway as selection changes, is there a change in that pathway?
You would hope so. And I think that is the reason why hearing aids don't work, right? Hearing aids, it's a bottom-up amplification. We want to have the top-down signal. We want the control signal back.
Yeah. But that would imply that even though, if it really is a generic selection mechanism, then the mechanism for going down to the feature level is generic too.
That's it could be. So there would be a lot of efferent pathways that are not mapped out systematically. I think the hearing literature has always been the afferent pathway, the bottom-up pathways. How the top-down pathways going all the way to the outer hair cells to tune specifically the cochlear amplifier would be the interesting part, right? It's hard to access to that data.
In the auditory scene analysis, I mean, you're saying you're using spatial cues, or you're evaluating spatial cues and pitch cues or harmonic cues. So another big cue is sort of grouping. I mean, it's onset and offset cues. I wonder if especially for things that are not harmonically structured. So I was wondering if you
That's the huge feature space, right? The onset tends to be more important than offset, but there are people that talks about how the change in the scenes between the onset and offset. So there's studies that Maria Chait over in UCL specifically studies the transient onset offset. To me, I think onset is just again another huge cues. What exactly are we doing in terms of as an organism to try and solve auditory scene analysis, right? Is to make sense of the world in terms of things that you care about, whether the lion is coming over from the right as opposed to some sound coming in, the roaring sound and all these frequencies. It's important for you to solve that problem. So you use all the cues. Onset is great because as a sound emulating in space, if they all come from the same source, they will have the same onset. They will all have the same facial relationship. And I bet that our brain capitalizes that information.
Not only that, typically offset is masked by reverberation, and it is way more basically fuzzy than the onset.
That's true. So onset is much more reliable. As a great Bayesian brain that we have, we would weight onset as a greater evidence than offset.
Yes, so the same in sound source localization.
Yes.
But there's cues for onset and offset that aren't affected by reverberation at all, which is say, watching someone's mouth or something like that.
True.
And I mean, we are just beginning to look at it, but it's very likely that there's crossover of these signals that can help you do this attentional gain that don't even come from acoustics.
I thought we were just talking audio here.
So for this huge literature on auditory scene analysis, books, all coming here, they talk about all kind of cues. Is there no attention injected in this at all?
Oh, no, I do not want to convey that at all. No, especially I just checked the box that this is going out. This talk is available to everyone. No, this is not.
So people already model attention.
There are many studies in the past that talks about attention. There are great debates on whether scene analysis is pre-attentive or not. Does attention matter when you segregate stream? There's a great long debate since early 2000. There a lot of studies before, I think would be fair to say, is from EEG, event-related potential studies. They answer different questions. I think that from a network approach is quite new, just because the MEG, EEG, stringing all these things together took a while to develop that technology. So in terms of how the auditory cortex communicates with the frontal eye field, I could imagine I could say that maybe, as far as I know, there are no other literature that talks about that out there.
How about in CASA community, computational auditory analysis? They have a group of people today. To what extent?
I know that, like Martin Cook, there are a lot of, of course they care about auditory grouping, right? But one thing that it turns out that from my doctoral thesis, a lot of CASA people think of, you have to phrase it in a probabilistic sense, right? Each time frequency pixel, it's either belong to speaker 1 or speaker 2, or whichever speaker it is.
How's the attention coming to all this?
So this is the interesting part. You can measure the same thing in a human. It turns out that they don't have to add up. There's no reason for a subject to think that each time frequency pixel has to conserve probabilistically. We phrase that model because computationally we need that constraint. Turns out when you do all there are asymmetries in the auditory cues that actually we pass a scene differently as the problem statement that the CASA people phrase.
So is the attention mechanism was there when the child was born, or actually the child learns it?
We don't know. I know that colleagues in our department is starting to look into it. I can tell you that in visual scene analysis, Pawan Singha, who was actually a reader of my dissertation back at MIT, he goes to India and looks at a particular group of children where they have this disease. I actually don't know what the technical term is. Basically, it's really hard for them to see, but at one quick procedure, they can fix the vision. So they can look at, when they can't do scene analysis to when they can do, and how quickly it is, and is it innate. So from the visual literature, there's this literature that can point us to the innateness for us to do this. In auditory scene analysis, unfortunately, we don't have that information yet, as far as I know.
Some people are just more absent-minded than others. Do you think that's because of genetic reasons?
This is the spouse notch filter that he was alluding to. Yeah. I'm pretty sure I don't need to test that. That's got to be universal.
That's good. Okay, thank you very much. So we do have, I'd like to
Thank you.